So moving to the next, we have that is bacteria with exotoxins. Continue. And bacteria, that is the bacteria that lyses cell membrane, that is Clostridium perfringens. Toxins produced is alpha toxins. The mechanism, talking about mechanism, that is phospholipase lecithinase that degrades tissue and cell membranes. Degradation of phospholipids, that is myonecrosis, gas gangrenes and hemolysis. Double zone of hemolysis on blood agar. Now Streptococcus pyogenes, toxins produced is streptolysins O, protein that degrades cell membrane. Lyses RBCs contribute to beta hemolysis. Host antibody against toxins ASO used to diagnose rheumatic fevers. Do not confused with immune complexes of post streptococcal glomerulonephritis. Now subrantigens causing. Now talking about super antigens causing shocks, that is Staphylococcus aureus. Toxins produced is toxic shock syndromes. Toxins, that is TSS1. Cross links beta regions of TCR to MSC class 2 on APCs outside of the antigen binding site. Overwhelming release of interleukin 1, interleukin 2, interferon gamma and tumor necrosis factor alpha, that is SOC. Also, that is Streptococcus pyogenes, that is erythrogenic exotoxins A, have the same mechanism. Now toxic shock syndrome, that is fever, rash, shock. Other toxins cause scalded skin syndromes, that is exfoliative toxins, and food poisoning, that is heat stable into enterotoxins. Now toxin shock like syndromes, that is fever, rash, shock, scarlet fever. Now moving up to the next, that is in talking about endotoxins. So endotoxins can cause, that is edema, nitric oxides, DIC or death outer membrane, then alpha necrosis, that is tumor necrosis factor alpha, O antigens, that is plus core polysaccharides plus lipid A, that is extremely heat stable, interleukin 1 and interleukin 6, neutrophil chemotaxis, shock. Here we can see about enterotoxins, that is not enterotoxins, that is endotoxins, that LPS, that is lipopolysaccharides, found in outer membranes of gram-negative bacteria, both cocci and rods, composed of O antigens and core polysaccharides plus lipids A, that is a toxic component. Neisseria have lipo oligosaccharide. So this is exceptions, that is all other bacteria, that is gram negative bacteria, outer membrane have outer membrane leaflets have, that is lipopolysaccharides, but Neisseria have lipo-oligosaccharides released upon cell lysis or by living cells by blebs detaching from outer membranes versus exotoxins, that is which is actively secreted. Three main effects, that is macrophage activations, TLR4 slash, that is CD14s. Now complement activations and tissue factors activations. So these are three effects, that is macrophage activations, complement activations and tissue factor activations. Here it is given, that is endotoxins, that lipid A components macrophage activations lead to release of interleukin 1 and 6 causing fevers. Tumor necrosis factor alpha causes fevers and hypotensions. Nitric oxides, that is, cause hypotensions. Complement activations leads C3A and C5 activations, that is, C3A cause histamine released, that hypotensions and edema. C5A, that is neutrophil chemotaxis. Tissue factors activations cause coagulation, cascades activations, that is DIC.